You may have heard a lot about DNA. DNA stand for deoxyribonucleic acid. Individual pieces of DNA formed the gene, and then collectively they comprise the entire genome. DNA was identified about 100 years ago by Friedrich Miescher, who was a chemist in Germany at the time. He was trying to get cell nuclei out of bandages used in hospitals, which are full of white blood cells that contain a lot of DNA. And he wanted to know what a chemical composition was. So he found that there was a lot of this stuff that he called, at the time, nuclein, and it took a very long time to realize that this nuclein actually was important for life. People thought it was just some kind of structural molecule that held the nuclei together. It is not until Watson and Crick figured out what the structure of DNA was, that people realized that it was a molecule that could carry information. Inside the cell, you will most commonly find double-stranded DNA, in which two strands intertwine to form a double helix. The most common form of the DNA double helix is also called B-form DNA. We can unwind the double helix so that we can see the chemical structure inside. Each strand is a polynucleotide, meaning the strand is made up of many individual units called nucleotides. A nucleotide has three components: the 5-carbon sugar, a phosphate group, and one of 4 possible nitrogenous bases: adenine, guanine, thymine, and cytosine. The nitrogenous base is always attached at the 1' carbon of the sugar. If we count from there, we can see that there is a phosphate between the 5' carbon of one sugar and the 3' carbon of the neighboring sugar. The sugar is called deoxyribose because it is missing a hydroxyl group at the 2' carbon which is present in ribose. Because of this, nucleotides in DNA are called deoxynucleotides. Nucleotides attach to each other in the DNA strand by phosphodiester bonds. The phosphate group of one nucleotide binds to the 3' oxygen of the neighboring nucleotide. Thus, we can see that the sugars and phosphate groups make up the DNA backbone. The carbon numbering is key to describing the directionality of the DNA strand, 5' to 3'. Looking within the sugars, there is an intrinsic orientation difference between the two strands. On the top strand, you can see that the 5' carbon of each sugar is on the left and the 3' carbon is on the right. The opposite is true for the bottom strand. Reading left to right, that makes the top strand orientation 5' to 3' and the bottom strand orientation 3' to 5'. Keep in mind that this double-stranded DNA is still a double helix, and we have simplified the representation by flattening and unwinding the helix here to better see the atomic structure. Although the nucleotides come together through covalent bonds in the backbone, the two DNA strands interact through non-covalent hydrogen bonds between the bases. Each base forms multiple hydrogen bonds with its complementary base on the opposite strand. Bound together by hydrogen bonds, each unit is called a base pair. The hydrogen bonding contributes to the specificity of base pairing. Thymine preferentially pairs with adenine through 2 hydrogen bonds, and cytosine preferentially pairs with guanine through 3 hydrogen bonds. Thymine and cytosine are called pyrimidines, characterized by their single ring structure, and adenine and guanine are called purines, which have double rings. The geometry of the AT or TA and GC or CG base pairs is the same, allowing for symmetry and base stacking in the helix. This mostly has to do with the distance between the backbones and the angles to which the bases attach to the backbone. Other base pairs, like GT for example, do not have the same geometry, cannot form strong hydrogen bonds, and disturb the helix. The double helix structure of DNA is highly regular. Each turn of the helix measures approximately 10 base pairs. In addition to the hydrogen bonding between the bases, the stacking of the bases also stabilizes the double helix structure. These pi-pi interactions form when the aromatic rings of the bases stack next to each other and share electron probabilities. The regularity of the helical structure forms 2 repeating and alternating spaces, called the major and minor grooves. These grooves act as base pair recognition and binding sites for proteins. The major groove contains base pair specific information, while the minor groove is largely base pair nonspecific. This is because of the patterns of hydrogen bond acceptors and donors that proteins can interact with in the grooves. In this way, the DNA can be acted upon in either a sequence-specific or non-sequence-specific manner, allowing proteins to position themselves correctly in the genome to carry out their designated tasks.